Good day. This is CSC239, Scientific Computing Applications. The day and date is Tuesday, 21 October 2025. Today we're going to change or break from our typical format when it comes to the assessments in a module. You have, certain certain regards, you have graduated from the walkthrough where we review the assessment live in class. We take time to do that, and then we post the reconciliation. What we're going to do for this module, the quiz has now been posted and it's online and it's available. It's due by 3 p.m., the start of October 23rd. So by this time, Thursday, everyone needs to have completed their first attempt of the Module 3 assessment. And that's important because what we're going to do is post the reconciliation in the assignments list. So the assignments, at the top of the assignments list, you'll see the 3.5 reconciliation. And I will have posted a separate recorded session where I walk through the items in the Module 3 assessment that pose the greatest challenge for students. So we won't take up any more time for the rest of the season when it comes to module assessments. We won't be taking time to review the assessment items in class unless, of course, students request that. If they say, hey, there was an assessment item and three of us are scratching our heads, we don't know what's going on. So we've watched the review of the item analysis. So the item analysis of each module assessment, the first attempt, is simply going to be posted along with the reconciliation assignment. And it'll be a separate session recording so that we can conserve the balance of the time we have in our class. And you can work independently to check on things that you need to check. If you did really well in the assessment, obviously we don't have to spend a good part of our class reviewing items if you did well. So does everybody understand the expectation here for Module 3 assessments? Module 4 assessments, Module 5 assessments, Module 6 special topics assessments. Essentially, the reconciliations you'll do independently. Any questions?
So basically, the format of the quiz is the same. The only thing that's changing is the reconciliation is not during class time, it's on our own.
Correct. Well, the
Okay.
reconciliation where you correct your own mistakes, that's always been independent. But that's a great question. It's the item analysis that we walk through in class where certain items posed greater challenges for students. That will still be conducted. It'll just be conducted separately by me, and it'll be a recorded link, right? So 15, 20 minutes, and the link for that will be in the reconciliation. So, yes. You will see just a reminder posted above the Module 3 references. I'll post a link like I did here. You'll see, oh, come on now. You'll see a link that basically says, hey, go to the quizzes menu to complete this. It'll have the deadline posted. Click on the quizzes. That's what we'll add at the top of the Module 3 listing in content. Now, I did have the references folder expanded. One of the things that I wanted to call out to you is that as we've completed our review of concepts and methods in Module 3, I have updated the study guide and the addendum, and then added some content to the lecture notes. So you need to make sure you have the latest copy of these references in your review before you engage. And once again, that's due by start of class Thursday, 3 p.m., this Thursday. Any questions? Well, yeah, you'll also find the recording for the item analysis inside the Module 3 session recordings because that's the final review for each module, the module assessment item analysis. Okay, so moving right along, Module 4 is broad and rich. And so I wanted to start to review certain things in Module 4. We are going to dabble a little bit. So what will happen is that for the rest of the season, we'll be offering some optional assignments, or as you might call it, additional or extra credit items. And we would normally post these here inside the final exam and other module. But we want you to go ahead and keep the coding skills and knowledge fresh. And so we're going to supplement modules 4 and 5 with optional coding. I'd call them explorations, right? We want you to explore Python as your time permits, just to keep things sharp. We do need to focus on specific topics in modules 4 and 5. And we may decide to go ahead and roll the special topics into module 5 this season. It all depends on our timeline. But the goal here is to complete module 4 by the end of the first week of November. So we're going to start introducing topics, and then we'll be engaging the assignments and solutions. And by the time we start class on 4 November, we will be posting the module 4 assessment item analysis. You'll have to have the module 4 assessment first attempt complete by that time. And then that'll leave us plenty of time for module 5 and some special topics. Really cool stuff. We're saving the best for last. So if you have copies of these PDFs on hand, just be sure that you discard those and you get a fresh copy of the latest version for module 3. Let's continue. So as we said, module 4 is broad and rich. There are actually 4 main topics, 4 primary interests, 4 critical skill sets, kind of like the Three Musketeers and D'Artagnan, right? You can't really have the Three Musketeers without D'Artagnan. And so when we get into scientific computing applications, there are some concepts and methods, a common language. We call it a lingua franca, I think is the term, the Latin. Anyway, it means common language. And much of that language has to do with the with what we would formally refer to as scientific method. Now, you may have encountered specific review of the scientific method if you participated in undergraduate research with the ECS, you know, the Caribbean Scholars Program. But if you haven't, it, this is something that's often misunderstood by the general public. There's a couple of very plain, very awkward misconceptions about the science, about proper scientific method. And considering this is a scientific computing applications course, I would be remiss in my duties if I, if I didn't bring you up to speed so that you wouldn't be considered a novice, right? You want to use the proper terminology. You want to understand the misconceptions about the scientific method by the general public. And it's as simple as misuse of the terms like theory, right? So um, in hard brass tacks, some things you'll never be able to prove. And so you cannot basically create a theory on unproven hunches, right? So how many of you know the real story of dinosaur extinction? Tell me if you've heard this one. How did dinosaurs go extinct?
A meteor.
A meteor, an asteroid, right? Um, it's called an ELE for short, extinction level event. There was a giant cataclysm. There's actually geologic record that validates this. Those are things that we can measure and observe. Okay. And here's something that they recently measured and observed. True or false? Dinosaurs are reptiles.
True.
Yes, dinosaurs are reptiles. Technically speaking, technically speaking. But in the later stages of the era, the age of dinosaurs, another thing that we've been able to observe that there is fossil record that verifies true or false, reptiles are cold-blooded. They cannot regulate their own body temperature. I mean, have you noticed that reptiles like to sun in the warm, in a warm, sunny surface, right? So that's because reptiles cannot regulate their body temperature. These are facts. I mean, these are things we can observe. There's clear evidence, there's proof of this, right? But the fossil record in recent years, what we've discovered is that there were very large dinosaurs, and if you look at the bones and you look at specific anatomy, when it comes to mammals, mammals are what we call warm-blooded. Some of you have had biology, right? Some of you are biology majors, yeah?
Yeah.
Okay. So this just in, there is fossil record for selected dinosaurs that tends to track with the kind of bones and vascularization that we see in warm-blooded homeotherms, mammals, right? And the other thing that they discovered is that, dinosaurs are huge, right? So the simple truth is the surface area of a large animal increases by a square of the dimensions of the animal, but their body mass increases as a cubed function because of the mass. Okay? Does everybody understand what I mean by squared versus cubed?
Yeah.
Okay, so smaller animals would have more surface area for their skin, right? They would have more surface area given their body mass, but the larger the animal, if you've ever noticed, elephants have to do a lot to cool themselves, and elephants have to cool themselves, and rhinoceros and a whole variety of very large animals, they have to cool themselves more so than smaller animals because they have less exposure in skin. Now, what I'm saying is that the very largest dinosaurs would have much less skin to vent their body heat, so they'd have much less exposure to the decline in temperature, right? And so there are some scientists that have studied other reasons why dinosaurs have gone extinct. Here's a good one for you, okay? Now, you're probably saying, okay, things are getting to be a bit of a wild card. Last week last class we talked about auctions and we went off into this thing about auctions. And now we're talking about the extinction of dinosaurs. Just bear with me a bit here. It turns out that when dinosaurs were roamed the earth, the climate in our planet was different. And basically, there was a lush rainforest with lots of leafy ferns that were coated with oil and wax. Put another way, it was kind of like a Caesar salad with olive oil dressing. And there are scientists that now believe, based on the fossil record and from evidence that we can observe, right? That essentially when the asteroids hit, yes, there was like a nuclear winter that resulted. So the skies clouded up and the sun went away and the temperature dropped and then all sorts of snow and ice started to happen. But remember, we've since discovered that there are a certain class of dinosaurs that, well, body temperature wouldn't be as great a challenge as we once thought. Body temperature would be less of a challenge for some of the recent discoveries when we talk about dinosaurs. Now, we do have a biology PhD on the Albert Einstein campus, and his specialty is, he's in the marine biology department, but he's a paleontologist, so he has a lot of interest in dinosaurs, and he's likely to call out some of the things I'm sharing with you to be a little out in left field. And I just want you to know that's okay. But essentially, um, what's happened over the last few decades is that they discovered, wait a minute, the temperature change, they didn't freeze to death. Some dinosaurs didn't go extinct because they all froze to death suddenly and there was an ice age. No, it's more that the climate cooled And they didn't have the leafy ferns with oily coatings and waxy coatings on the leaves. They didn't have their Greek salads to munch on anymore. And what happened instead is that the cooler, drier temperature gave rise to dry grasses like straw, okay? Straw. And let's face it, there are some mammals that have an evolved digestive system, four-chambered digestive system that can handle that. But dinosaurs, yeah, no. All right, so they were eating dry grass and straw and hay, and that was a huge change. And that created all kinds of problems with their basic digestion. They became constipated, and this became another strike against them, another nail in the coffin. But here's the thing. How many humans were around to observe directly whether or not dinosaurs became constipated? I'm just asking the question. From what we know of historical record, were the earliest humans around when dinosaurs roamed the earth? Yes or no, from the historical record that we have for observation. What do you know about that? It's not a trick question. Hello? Um, can I get somebody to unmute your microphone and let me know
I,
whether or not you're still here with us?
we still here,
You're
but
still
I
here.
don't answer
Okay.
that.
I'm sorry. Did the topic totally turn you guys off? Dinosaurs dying out and going extinct because of constipation? It's kind of a crazy thought, isn't it? Was anybody around? Were humans around? I mean, everybody knows the answer to that pretty much, right? Humans didn't come along until much later, according to the scientific record. So the simple truth is, we don't have a way to observe directly certain things we study. And that has to be qualified properly if we have integrity, if we speak with credibility, if our results matter. And if we don't qualify things properly, well, um, it's like it's like the tabloids that you see in the grocery store. There's all these crazy magazines and newspapers um, that people read sometimes just for entertainment. It's a nice story, but there's no real truth to it, right? And the thing that I want you to know is that we talk about the computing aspect of the scientific method more so in this course and the way we work with data, but the real truth is there's a piece of the science method that warrants a closer look, right? So in case you hadn't you hadn't really been introduced to proper scientific method, we have this thing called a hypothesis. What have you heard about a hypothesis?
It's basically like a prediction of a study, what you think might happen.
Yes, there is something about prediction. Yes, thank you. Hold that thought. Okay. Now, we've told you that when it comes to quantitative analysis, that has to do with numbers. And then when we talk about qualitative analysis, that has to do with patterns and relationships, what we would sometimes call or refer to as correlations, right? There's a pattern, and the data speaks to a predictable a predictable outcome. So the word prediction is embedded, it's just dripping. The idea of prediction is an integral part of the scientific method. And a lot of times, the experiments that we devise to collect experimental data, essentially, people tend to think that the goal of the experiment itself, the goal of the experiment itself is to prove the hypothesis. And no, that's not it exactly. A hypothesis is an informed conclusion or decision. It's a prediction. It's an informed prediction based on the pat. All right, so there's initial observations and initial and preliminary research. Okay, so you have this period in the early stages of scientific method where you're curious about a thing, and there's preliminary research that you would do online or in the library, and you'd see what other people have been doing in that area. But real science is exploration. Real science is discovery. It's not going to the library and doing a study of what everybody else has studied, which is what a bibliography is all about. You have a specific topic and then you compile the research for a given interest, and then you look at all the cool stuff that everybody's done and you report on what other people have reported. Let me say that again. You've reported on what other people have reported. Significant, yes. It has value, to be certain, but that's not where the real action lives and breathes. That's not where the real excitement happens. The real deal about scientific method is that if we use it properly, we discover new things. We understand what was previously unknown. And I got to tell you, if you've ever done science properly, our science course labs are really more like cookbook exercises. Now, I'm not criticizing our science labs, but have any of you had seen a chemistry lab where you mix vinegar and baking soda? Has anybody ever seen that?
Oh yeah.
Uh-huh. So you mix vinegar and baking soda, and what happens?
It kind of starts forming up and
Yes.
thing.
Yes, it bubbles and it produces carbon dioxide gas and it neutralizes the acetic acid, the acidic part, which is a lower pH, right? So pH numbers that are more acidic are lower, and pH numbers that are more alkaline are higher. I hope I'm touching on things that you're familiar with already. And the thing is that what we do is we subject our students to so many what I call cookbook labs, experiments where, well, we know the outcome. And what it really is practice. It's practice to understand the steps of the scientific method, especially the parts that have to do with measurement and pattern recognition. When the baking soda is dissolved by the acetic acid and it produces the carbon dioxide gas, the gas is lost to the atmosphere around us. So there's a decrease in mass. Okay? Does everybody understand what I mean by that? It's like you have so many milliliters of vinegar and it has a mass of, close to 1 milliliter per 1 gram per milliliter, typically speaking, right? Did you know that 1 gram is technically 1 milliliter of water at 4 degrees Celsius? Precisely, exactly. I mean, to a point of being perfect. if you measure water, 1 milliliter of water, and the mass of 1 milliliter of water at 4 degrees centigrade Celsius, right? It's exactly 1 gram of mass. That's pretty profound when you think about it. I mean, because we use water in all kinds of contexts, especially biological systems and in chemistry and so on. Anyway, a big part of acetic acid or vinegar is water, right? So we can measure the mass of the vinegar and we can measure the mass of the baking soda before the reaction, and then we can we can deduce, right? We can deduce from the mass that disappears how much of that was carbon dioxide that was a product, right? We have reactants and we have products. The point I'm trying to make is that we practice cookbook labs where we know what's going to happen. And the idea is for you to practice the experimental methods like measurement and the calculations required, and of course, the reactants and how they, after the reaction, you have products and so on. And it gives you great exposure to the essentials. But let me tell you, I've taught science in another life, like honors physics and chemistry, college prep physics and chemistry, 11th and 12th graders. It's a long story, but in another life, a long time ago, in a galaxy far, far away, I did that. I used to hate labs like that because it wasn't about the scientific method. There wasn't anything open-ended. We weren't exploring things that nobody knew about. Half of the spark of human discovery, it boils down to your own creativity and your own curiosity about things that you'd like to know more about. I mean, do you just want to memorize a bunch of facts? Or do you want to discover something that nobody else knows? And if you're credible, if your methods are sound, if you use the right process, then other people can repeat the experiment. That's one of the things that's most important about our discovery methods is that when we're applying the scientific method, we have to keep records of the specifics for how the tasks are performed, faithful records to capture. And this is where scientific computing and data come in. I mean, we used to have lab books and the pages looked like grid paper. Does everybody know what I mean? It's not a composition notebook. It's all chart paper, right? X and Y axis overlaid on the pages instead of just composition lines for English where you write. Or is that before your time and you don't know what I'm talking about? I promise you, I won't start talking about recording tape and LPs and stuff like that, 8 tracks and things. I mean, you're still old enough to know what a CD is, right? Please tell me.
Oh yeah.
Okay. All right. Above all, any discovery worth sharing in the world must be repeatable. And that's where the data processing methods and statistical analysis. So when I talk about capturing the data and keeping a meticulous record, that's the measurement data capture. And then once we have the data that's captured, the measurements that are captured, the inputs from sensors and so on, then we analyze the data. And there's two basic worlds in statistics. One of them has to do with descriptive, and the other one is inferential. One of them is quantitative. It's more about the numbers, the patterns in the numbers. That would be descriptive analysis. Okay. So if we say descriptive statistics, descriptive statistical analysis is quantitative math that helps us describe the patterns of the data that we've been collecting. The qualitative, the inferential statistical method, inference. Inferential statistics is about we take the pattern recognition to a whole different level. And what we're identifying are relationships between data elements, right? And then we have these things called dependent and independent variables. Has anybody ever heard of those terms, dependent and independent?
Yeah.
Okay. So here's a question for you. And this is something that throws people all the time. Okay. All right. So we've said that there are 4 sections in Module 4. What a coincidence. It's Module 4, and there are 4 main sections. And we want you to describe the critical components of the scientific method and their relevance, utilize macros and advanced features, pivot tables to process data consistently. That has to do with numerical data and descriptive statistical analysis. Contrast the 2 kinds of input data used for statistical analysis. So we can look at the world of input data and break those down into 2 main categories. So, not to bore you, but we have 2 main categories of pursuit here, qualitative and quantitative. And when you look at the quantitative, right, we're looking at more like the numbers. That can be broken down into, if you're dealing with the numbers, that's statistics. The quantitative analysis that we perform can be broken down into descriptive versus inferential. And you're probably familiar with descriptive statistics because of the kinds of things that you've heard. Has anyone ever heard of like mean and mode and standard deviation, min, max? Of course you have. You've had math classes, right? Has anybody here had statistics yet?
Yeah, it's like 2 years ago.
Okay. So you're probably going to think I'm a novice when it comes to statistics, and that would be a correct statement. Compared to what you engaged and encountered in your statistics course, I'm going to be simplifying some things, kind of distilling them down into the core concepts you really need to know and the core methods. Let's talk about standard deviation as a simple descriptive statistical analysis method. Can everybody see this screen?
Yeah.
Okay, so when scientists and mathematicians begin to investigate the world around them, and they start to measure all manner of items and organisms and phenomenon in nature, oftentimes they begin to understand that some of the natural things that evolve, there's this thing called a bell curve. When you have a population of subjects and you perform certain measurements, you find out that there's uh, more of an average number among the population, but then there are a few exceptions, and then there are even fewer rare exceptions. Do you understand what I'm talking about? Let's go back to this. Have you heard of a bell curve before?
Yeah,
Let's
I've heard
just
about it before.
let's just use a simple example. Let's talk about body mass among humans. Are you with me? We're just talking about the amount of body mass that the average human carries around. Is everybody with me? Let's say that we measure the body mass of an average human population. What we find is that um, there's a huge number. So if the y-axis is talking about the numbers of subjects or organisms or humans, right? The number of humans, and and this talks about their body mass. So it's I'm sorry, this is the y-axis. Did I say x-axis before? I think I did. The y-axis would be um, the numbers. So the quantity of individuals. Let's say that the average, what would you say the average range of normal body weight is for most people? I'm talking about grown adults.
Um,
Well, let's talk about very very healthy, very slender, and somewhat height challenged individuals. Okay. Um, would it be fair to say that among the population of adults, now let's talk about the U.S. Virgin Islands, okay? That among the population in adults, most adults in the U.S. Virgin Islands would would have a they'd have a weight that would run between 100 pounds and like 220 pounds or 200 and what do you think? 250? 100 to 250?
I see, yeah.
Okay. And the truth is, there's probably a greater number of adults in the U.S. Virgin Islands that aren't as light as 100 pounds, right? So there aren't as there there are fewer people that are closer to 100 pounds, and there are fewer people that are closer to 250. And the truth is, there's a whole lot more people that are like 130 to 175. Would you agree?
Yeah.
Okay. So in terms of descriptive statistical analysis methods, variance and standard deviations is a very helpful method because it helps us understand the distribution of variations and deviations from and what's the average, what's the min and the max and all this kind of thing. And if we have adults that are like 100 to 120 pounds, I'm just saying, let's pretend, are you with me? Let's pretend that this graph, the x-axis is increasing weight and the y-axis is numbers of individuals, okay? I mean, that's not how it's labeled. I'm just kind of making it up as I go. Okay. So, and then we have fewer people that are like from 175 to 250. Would you agree? We would call that like a standard deviation. Now, I'm just using this as a quick shoot from the hip example, but most people would run from like 130 to 140 to 175, and that would represent 68.2%, a standard bell curve. So statisticians, there's a mouthful for you. statisticians have discovered that when it comes to a standard bell curve, within the most common range of values, it includes 68.2% of the entire population. Okay? And okay, this is just a general example, again, shooting from the hip. If you had adults that were like 100 to 130 pounds, and then you had other adults that were 175 to 250, right? That would be a whole deviation. It'd be a level of deviation, right? It would be another level of deviation from the norm. So you have these terms that they use where you talk about, and I want you to notice the further out you go, now you have some adults that are what, 60 to 75 pounds, and you have some adults that are what, 260 to 320 pounds. And then at the far end, you have some adults that are like 50 pounds, and you have some adults that are like 400 pounds. You understand what I'm talking about, right? One of my goals here is for you to know, like the back of your hand, the method that you use to calculate variance and standard deviations. And we're going to get into the weeds a little more on this in our next class, but I want you to, and there's some very cute, very complex, very fancy ways of describing how this works, but I want to give you a simple example to demonstrate how to calculate variance and then standard deviation. Okay? So I want you to pretend that you're measuring the temperature of water samples in a store where you have the refrigerated produce section and the freezer section, and then you have other sections, right? Let's just pretend that there's a wide range of temperature there, okay? And let's pretend that it's also they have a deli, and deli, they heat stuff, and you can like make take-home plates. So there's cooking involved, right? So we have heat lamps. So let's pretend this is Celsius. Is everybody with me? Just a simple example. I'm in a grocery store with a deli, and it has everything from like frozen food section. And I'm walking around with a thermometer, and I'm taking random temperature samples at novel places, unique places around the store. Is everybody with me? I was thinking I'd be able to end this class in like 3 minutes flat if somebody responded, but I guess, can somebody unmute your microphone so I know you're still there?
We're still here.
You're still there. So what do these numbers represent? What type of measurement did I say I was doing in the store?
Celsius.
Celsius, temperature, right? Whole numbers. And let's pretend I make 6 measurements. Is everybody with me? And I'm just going to use this as a simple example. I find a place in the store, 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, 40 degrees Celsius, 50 degrees, and then 60 degrees in the deli where we have heat lamps. We're keeping food warm. Everybody with us? Okay.
Yeah.
Okay. Now, do you or do you not know how already from your math classes that tortured you? That's an unfair disparagement. My apologies to every math professor in the College of Science and Mathematics. You do know how to calculate the average of those 6 measurements, do you not?
We do.
Okay. So you add them all up and you divide by what?
You divide by 6 or the amount they have.
The number of numbers. You divide by the quantity of numbers. We have 6. So if we add all these up, we get a total of 210. We divide by 6, And that gives us an average, which has another fancy M term. What's the fancy M word for average? Is it mean or median?
Mean.
It's mean. What's the difference between mean and median? Does everybody know what that subtle difference? There is a subtle difference. So mean is correct. Mean means average. Okay. Median would be a particular measurement that represents the midpoint between the lower end of measurements and the upper end of measurements. And it may or may not have anything to do with the average. Okay. Do you see 35 here? Was that a measurement? Let's pretend for just a second I had a seventh measurement and it was 35 precisely. Well, what's the difference? I have three measurements below 35. I have three measurements above 35. So 35 as a measurement, if I had a seventh temperature measurement and it was exactly 35 degrees Celsius, which gets to be pretty close to human body temperature, right? Right? Human body temperature, 98.6 degrees Fahrenheit or 37 degrees Celsius. I think that's correct. Okay, so that would be the median, but if we do the calculation to determine average, it's also the mean. Everybody with me so far? Please say yes.
Yes.
Okay. Now, here's the fun part. We want to calculate a descriptive statistical quantity called variance. And variance is a part or an essential component of the grander calculation we call standard deviation. So in order to reach the goal of calculating standard deviation for a population of measurements, we first have to determine the variance of those measurements or data collected. And what I want you to do is I want you to notice the fine print here. So we have an average here, correct? 35? Yeah? And then I want you to remember these measurements, 10, 20, 30, 40, 50, 60, yeah? So if I take the average and I take the first measurement, what was the first measurement? 10 on the previous page, yeah?
Yeah.
So if I take the mean and I subtract the lowest measurement, the difference is 25. I square the difference. What's 25 squared? 625.
625.
There we go. Now, what's the next measurement in that series I showed you on the page before? It was 20. We do the same thing again. I take the average and I subtract 20, which is the next measurement in the population, in the number of measurements that were conducted or performed. The difference is 15. What's 15 squared? 225. The next measurement was 30. What's 35 minus 30? 5. What's 5 squared? 25. Now, what's the next measurement? 40. Now I have 40 minus 35. What's the difference? 5. I square it. What's that quantity? 25. Now I take 50 minus 35. That's 15 squared. I have 225. And finally, I have a top measurement, the largest measurement, the greatest value, the max value. 10 is the min, 60 is the max, right? We know what the median is, we know what the mean is, and now what we've done is we have squared the differences between the measurements and the average. We've squared the differences between the measurements and the average. And now, this is kind of like an iteration. If we add all these numbers up to get, we get this, 1750. How many quantities did we measure? 6, or how many did we calculate? We calculated 6 squares of the difference. Notice my words. We calculated 6 squares of the difference. And the total for that is 1750. Now, what's the average? Right? If I have 1750 and I have 6 of these numbers to work with and I divide by 6, it's like an average of an average, but there's a square difference in the middle. Does everybody see what I'm doing here? That's called variance. So when we divide 1750 by 6, we get a variance of 291. And then here's the fun part. Everybody knows what a square root is, right?
Yeah.
What's the square root of 64? Quick.
8.
8. Yes, thank you very much. What about square root of 81?
9.
Square root of 36?
6.
A square root of 9?
3.
I'm not going to trick you, am I? Okay, so don't you love it when things are sensible and easy? Standard deviation is the square root of the variance, which in turn, now I want you to notice what we did here. We calculated the square of the difference between the samples and the average for the samples. And then we added all those together and got the average of that. That gave us the variance. How widely are data measurements? That's the variance for the data that we measured. And then we take the square root and we get 17.06. Now let's come full circle. 17.06 represents the standard deviation for a bell type curve. And we're just talking about the recurring pattern of natural deviations found in nature. We find these in Mother Nature, right? And so you see here, what's the number? 34.1%, right? That is a standard deviation from what does this line represent right here? Right in the very middle. That's the median. Okay, we were talking about mean and median. So I wanted you to have context when we start calling out this stuff on the charts, on the graphs that we're showing. This is the single measurement that represents the midpoint between all of the lower measurements and all of the higher measurements. All right. Or is that the mean? I forget. I'm going to look that up. Well, it's the midpoint. And when it comes to human beings and their body mass, there is a certain percentage that we call the standard deviation. Some less and some more. Okay. What we just calculated was like half of that. So what we have is a graphical representation that's much thinner than all right, so if you have a perfect bell curve, and this is called a bell curve, then what you have is 68.2% of the samples or measurements or items fall within that, some a little below and some a little above, within that range of the midpoint. or median, right? Now, in the example that we showed you, instead of 30, what do we say, 34.2? This is 17, right? So when we're looking at a graph of that, instead of being shaped like this, it's going to be shaped more like this red curve. It's squished more together, and a standard deviation would be split like thinner. And so it wouldn't, this is sort of like, if you look at these representations, right? Different populations of individuals or organisms, right? Or items that we're studying, or subjects, or data collection points, or sensor readings, right? Does everybody understand what I'm saying here? Sometimes that curve is very flat and sometimes it's very steep. And what we have here is something that's halfway between the red curve and this curve here. It's the standard deviation is 17.04. In fact, if it was 17.05, it would be exactly half of this 34.1. Does everybody see where I'm coming from here? Okay, now what I want to do is to hit the pause button, and I want you to watch the segment of this recording where we walked through how to manually calculate the average or mean and then calculate the differences between the mean and the measurements and how they were squared and then all added together and then averaged again. That's variance. And then we square root that to get the standard deviation for a population or for a number of measurements that we're analyzing, okay? Actually, I said 17.06. Would have been 17.04 would actually be really, really good. Really, that's very close to being half of what we showed you on the chart. All right, what we're going to do is hit the pause button here. Does anyone have any questions, comments, or observations about what we've shared about our first review of the concept and methods we affectionately know as standard deviation and variance? You want to memorize the steps that we took to do this, and you want to be able to do it by hand because it needs, otherwise, if you don't do it by hand and you don't learn it that way, it's just another statistical method and it doesn't have meaning. When you see live examples of what it is and how we came to that result, I mean, I used an example of a grocery store, but could be something else. Could be the mass of leatherback turtles in a population of leatherback turtles in your neighborhood, okay? And the standard deviation for the measurements of their body masses in kilograms could be significantly different than the perfect bell curve. And aren't you glad we have computer programs that do this for us automatically? I'm just saying. Okay. Any questions, comments, observations before we clear today? Good luck with your first attempt of the Module 3 assessment. Remember, we're not going to review, but if you don't have your first attempt log by the start of class on Thursday, well, you won't have anything to reconcile. So please be sure you log your first attempt. Thanks for joining us. Until our next session, be safe, take care.